Hi everyone, my name is Anna Pichugina. I'm from Lomonosov Moscow State University. The title of our study is Holistic Face Processing Eye Movements Analysis. There is a great interest in the topic of facial perception in the scientific literature. Different lines of research have shown that faces are special stimuli. Eye tracking is one of the most effective methods for studying the mechanism of facial perception. However, in studies with eye tracking, there is a problem of data heterogeneity of results. This, in our opinion, may be due to several reasons. One of them is different processing eye tracking, different ways of processing eye tracking data. We made the third point the subject of our research. Eye tracking data processing is most often associated with the identification of areas of interest and the analysis of indicators within them. However, the authors identify areas of interest according to their personal ideas. There is no single standard. So you can see some examples. In some cases, the area of nose bridge is combined with the area of eyes. In some, it is combined with the area of nose, and sometimes it is analyzed as a separate area. We decided to investigate how different markup options will affect the results. The experiment involved 57 people. SMI IVX eye tracker was used to record eye tracking data. For the experiment, 10 photographs were selected with neutral expressions. For each photo, we made several distortions. And then for this, every photo, we made an inverted version of this photo. We used inversion because it's a classical technique that is believed to destroy holistic processing. It is very oftenly used in studies of facial mechanisms. Photos of upright and inverted faces were presented pseudo-randomly. After each photo was presented for 2 seconds, the participant had to assess the attractiveness of this face on a scale from 1 to 9. And then we used 3 types of area of interest identification. The first variant included the area of nose bridge as a separate area. In the second variant, we divided this area between the eyes, and in the third variant, we combined it with the nose. On these graphs, you can see the dwell time in the areas of interest when people looked at the upright and inverted faces in 3 variants of areas identification. You can see that in the first variant, there are differences in the area of nose and nose bridge. When we divided it between the eyes, we still can see the difference between the areas, between the dwell time in the area of nose. But in the third variant, these differences disappeared. It is not surprising since in the first variant, the differences between the nose and nose bridge were had different directions, had opposite directions. So when we combine it in one zone, we have no difference. We decided to examine individual strategies. We used the indicator of ratio of the average fixation duration to the average saccade amplitude recorded during the examination of the upright faces. And we got 2 groups, one with a static eye movement strategy, another with a dynamic eye movement strategy. And we examined the intergroup differences when observing upright faces. As you can see, in all 3 variants, we have significant results in all the areas, and they have the same directions. It means that the individual strategies are very strong, and we have to take it into account when analyzing perceptual mechanisms. We can make several conclusions. One is that in the entire sample, the presence or absence of statistically significant results in the dwell time depended on the method of identifying the areas. The second conclusion is that when conducting an intergroup analysis, we found no differences depending on the markup of these areas. And the third conclusion is that a more detailed type of identification of areas, the first one in our experiment, is preferable for analysis because the inclusion of this area to the area of nose or eyes ambiguously affected the results. Thank you for your attention.